of body length, and we can see this is quite common for keating nymphs that we have very distinct size groups. We have small guys, medium-sized guys, and large guys. What you can do then is use something called mixed test, which clearly distinguishes the cohorts from each other, and it's going to tell you the mean length of the animals in each size cohort. So this is an example from Svalbard for P. elegans. If we look at the data from the Canadian Arctic for the same species, well, maybe the size cohorts aren't as clear as we had in Svalbard, but we clearly see that there's a summer generation. We have very few individuals being born after summer. After, for example, September, it's very rare to find individuals less than 5 millimeters. So we can look at the growth then. If we follow the mean lengths of the different size groups in different months and subsequent months, we can look at their growth. We can do that for the small guys here, from when they're born. And you see that after 1 year of life, their length exactly matches the length of the medium-sized individuals, the cohort 1. And it's also important to say that individuals of P. elegans can reproduce when they get to around 1 year. They have the gonads well developed by that point. Right? They continue to grow for a bit longer, and we see that they become around 40 millimeters after just over 2 years of their life. What is very important to mention now is that in all of those size groups, the period of peak growth is the same. It's always in the summer, the same time that reproduction occurs. So we know that the growth is linked to the period of the phytoplankton bloom. And these data here are from a previous study in the Amazon Gulf in 2008, and they show that chlorophyll fluorescence definitely peaked near the surface between May and July. Okay, that was one species. I'd like to go back to E. hamata now. Also abundant, but it has a much more complex strategy, because what happens here is that there's 2 generations per year. We have one that's born in spring and summer, and then there's a gap, and then we have another one in winter, in contrast to the only single generation seen in P. elegans. We can also fill in some of the gaps here. We can look at the other size cohorts by assuming kind of reasonable growth rates between months. That was a bit more complex to do. Okay, now I've told you about how these guys are living. I have a lot more data as well on what depths they kind of prefer and when they're mature and stuff like this, but I'm going to go on to the next question now, right, which is, are they really the tigers of the plankton, as some people have suggested? Well, much more often than having prey in their stomachs, they seem to have detritus and marine snow. We see here individuals from last year's cruise of E. hamata and P. maxima. These were living individuals preying or feeding on green stuff, on green material under the microscope. Okay, so there's no prey involved there. If we look at what that green stuff actually was made up of, then there's lots of dinoflagellates and lots of algal species there. So what I'd like to do really is reconsider their role in the ecosystem, reconsider their feeding mode, and ask the question to you, are they the tigers, or should they really be referred to as the sheep of the plankton? It might be more that they're osmofilterers, so to speak, which means that they really just have their mouth open at all times, and they're taking in water at all times, removing the particles from it. And when they can feed on a copepod, for example, they may take that as well. So just to recap now, seasonal studies such as the one I've just talked about are key to fully understanding the life histories of Arctic zooplankton. The fact that we had a 1 year time series really allowed us to answer some questions that we wouldn't have been able to answer if we'd only sampled during one season. Although I don't have so much data on Parasgita or Pseudosgita maxima, most of my data is on Uchronia hamata and Parasgita elegans, but we have shown that they have very different life histories in the Canadian Arctic with respect to when they reproduce, for example, how many generations they have in a year, when they grow, and where they are in the water at different times of the year. My last point here is that there's very little evidence that these animals should be called the tigers of the plankton. But my next chapter will focus more on their feeding modes. And we're going to use an inverted microscope to see exactly what's in their guts and come back to that question. Okay, here are my acknowledgements and my references. Thank you for your attention. I'm sorry the slides didn't work how I'd planned, but if you have any questions, I'd like to answer them. Thanks.
Thank you. Yeah, technology sometimes is unpredictable. We do have time for one question. So we have for question is that if the Do you have temporal scale data? Is the productivity change now with time, especially with the climate change, with like the length of growing season and as well as quantity that is produced?
Yeah, this is a very important question. It's probably one of the key questions which affects zooplankton in the Arctic, because we think that the phytoplankton bloom is coming earlier, and in some places, due to climate change, the ice algal bloom may not be there anymore. And this is very important because it could affect the food available for lots of zooplankton. And now I'm showing maybe keeping nymphs. If the zooplankton is not there to feed on the phytoplankton when the phytoplankton is there, they could starve and they could have problems for the rest of the year. So we need to kind of think about those questions in more detail.
Thank you very much. We're moving on our next speaker, Bernabe Moreno from Científica del Sur University in Lima, Peru. And Bernabe is going to talk about spatial distribution of the macrobenthos of the Mackellar Inlet, King George Island, explanation integrating the systemic and trophic grouping approach. So Bernabe, the platform is yours. If you unmute your microphone,
Yeah, can you hear me now?
Yes, we can hear you, so you're good to go.
Okay, thank you. Well, greeting from Peru. My name is Bernabe Moreno, and today my function is to present to you some of my results of my thesis that I've called Spatial Distribution of Macrobenthos, and I've used here an integrated approach of the systematic and the functional grouping approach. In the Mackellar Inlet, I think I have the same problem of Jordan. So, well, the Mackellar Inlet is located in the Western Antarctic Peninsula, and this is considered a region of a very strong recent warming over the last 50 years. Also, this is an important region because, well, Julian Good has pointed out that in this region there is a high number of community types, and also it's one of the most sampled zones of all Antarctic. So there's been a really high importance in this zone, and also this warming is attributed